Okay, so our experiment focuses on dopamine and aggression. Alrighty, so first I'll start off with some background information to help demonstrate why this experiment is important. And it's important to note that the brain of the fruit fly and the human are very similar in terms of genes, neuromodulatory systems, and circuit organization. Therefore, studying the fruit fly can give us some insight into the human brain and see how affecting the receptors and neuromodulators can translate to behavior. And essentially, the three aims of our experiment were to map the neural anatomy of the dopaminergic neurons in the fruit fly, see the effects of downregulating the dopaminergic neurons and on aggression, and seeing the effects of upregulating the dopaminergic neurons. Okay, and so these are our 4 fighting chambers, and the fighting chambers are one's gonna contain flies that express channel rhodopsin to which will excite their dopamine systems upon having red light shined on them while they're fighting. Another one will contain flies that express halorhodopsin, which will suppress the dopamine system while yellow light is shined on them. And then the other 2 will serve as baseline and will have no light shine on them. The chambers are made from a petri dish with 4 glass slides glued together as the chamber. The lid's on top so the flies can't get out. In the center, there's a little hole with a cap on it where we can aspirate the flies into. And there's also fly food, and a decapitated female to release pheromones that'll hopefully incite the flies to fight. And directly after aspirating the flies into the fighting chamber, we'll begin recording for 20 minutes to see their aggressive behavior like approach, low-level fencing, and lunging. All right, and next Sheila will go more in depth about the crosses and neuroimaging.
Hello. So for this experiment, we did 3 different crosses using the binary GAL4 UAS system. This way, the GAL4 would be the enhancer and the UAS line would be the reporter. We use the same dopaminergic GAL4 line in all of our 3 crosses, and that GAL4 line expresses chromosome 3. We used we were supposed to use adult male GAL4 in all of our crosses and then female pupa in all of the UASs. But for our experimental group, we didn't have any pupa at all in our vials when we got them, so we had to flip it around. So two of our crosses had the adult male in the GAL4, and then they had female pupa from the UAS line, while our experimental group had female pupa from the GAL4 and then adult male from the UAS line. So for our experimental group, it's like this image show right here. We crossed a female pupa GAL4 with the adult male UAS YFP tagged light activated halorhodopsin channel fly that basically gets activated with yellow light. So like this offspring right here will like get activated with like yellow light when it's shown up on it. Our control cross was GAL4 again with the UAS enhanced GFP line, so that like when any type of light is shown upon it, like it wouldn't like activate or inactivate. Well, we wanted it to be like silenced. So yeah, that's why we use this enhanced GFP so that it wouldn't be affected by any type of light, and it would be there our control. Our third cross was to acutely activate the dopaminergic neurons. So we crossed the GAL4 line with the UAS red shifted channel rhodopsin flies, and they will get activated with like red light when shown upon them, and this basically like increases like the activity in the brain, and it makes the flies like more aggressive than like they normally will be in like the control. Neuroimaging. We use fluorescent microscopy to map out the neural circuitry of like the Drosophila brain. This image right here is like from the posterior of the decapitated brain, and this like whole brain is from a UAS GAL4 fly, and that GAL4 fly was from like the DA neuron, so it's like expressing dopaminergic like neurons in it. So right here, like in the image, you can see like the green, like being like green firing, like that's like the DA neurons like firing in like the brain. Um yeah, and we use like fluorescent imaging for like all of our experiment to figure out like how like DA neurons are being like transcribed, basically to like see how it's firing, like the inhibition and like activation and like the silencing of the neurons. So yeah, my group is gonna talk more about the whole imaging in the next slides. Thank you.
For our results, we see an increase in aggression in male flies and the overexpression of dopamine activity in comparison to the baseline expression. As you can see by the table, both fly E and F exhibit more offensive behaviors than fly A and fly B. There's especially an increase in both low-level fencing and lunging. In the underexpression of dopamine activity, we see a significant decrease in aggressive behaviors, as you can see in fly C and fly D. Once again, especially in low-level fencing and lunging behaviors. In the next slide, we have a bar graph to better demonstrate the data.
Okay, continuing with our results, we used a paired samples t-test to analyze our data, and we used that because we were looking at the differences in behavior as a result of varying levels of dopamine expression in male Drosophila. And as we can see in the chart, our data appears to show that there appears to be more occurrences of behavioral aggression as we increase the levels of dopamine expression, because you see in the underexpression of dopamine that there's no occurrences of low-level fencing or of lunging, but we see in baseline that there are more overall occurrences of aggressive behavior. And then we see in the overexpression of dopamine that there's even more occurrences of the approach, low-level fencing, and the lunging. So from that, one would think that our results show an increase in aggressive behavior as that result. But when we did the paired samples t-test, we found that there's no statistically significant change in aggressive behavior compared to the baseline as a result of the dopamine neuron over or under expression. And we did that with a 95% confidence. In conclusion, using the Drosophila melanogaster, also known as the fruit fly, as our experimental subject was a good decision because they make excellent animal models. And I say that because when you look in the diagram to the right, you see that they have a very short lifespan and life cycle where they only have a 2-week development, and their life cycle only lasts 40 to 50 days. And this was very helpful when we were passing our flies from the F0 to F1 generation because it helped us to do it quickly. And it also helped us to be able to carry out our behavioral analysis because we were using fight chambers and we had to use socially naive flies. So being able to produce those flies in a short amount of time was very helpful to the experiment, and it was not very consuming with time. And another plus to using the Drosophila was that they're relatively inexpensive compared to most other experimental animals like mice or monkeys that you might use for these types of experiments. So they're very inexpensive because of their size and because they reproduce fairly easily. And another thing is they only have 4 chromosomes, which made it like very easy for us to have genetic manipulation done because we had to use fruit flies with over and under expression of dopamine. And it was just very easy to do that since there's only 4 chromosomes to work with. And another plus to using the fruit flies was that they're easy to compare to humans. And I say this because they have a lot of homologous structures with the human brain. And this is very important in our experiment because since dopamine is expressed in multiple parts of the brain in humans and in fruit flies, it was helpful that the structures are very similar in the fruit fly and human brain because that helps us to compare their aggressive behavior to human behavior. And another pro and con to using the fruit flies was if you could see in this diagram to the left that like dissecting the fruit fly brain was a little bit difficult just because their bodies are so small. So that was kind of a con because using a bigger animal like mice or something like that, it could have been easier to dissect the brain and get a visual readout for their GFP and DAPI visualization. But the only thing is that could raise ethical concerns because it seems like the bigger the animal is or just the more complex their brain is, it could raise ethical concerns. So that's the pro part of using the fruit flies for the visualization. And yeah, another thing is them being so small, they're very easy to just contain in general. So storing them and just labeling them was very easy in our experiment. So overall, they were a good animal model to use.